Welcome back to our journey into the cell membrane. Now, if you thought simple diffusion was the whole story, you are in for a treat. Today, we're jumping into the high-energy, high-stakes world of active transport. This is basically cellular rebellion. It's how our cells defy the natural flow of things and, bend the rules of physics to build the very world within themselves. Let's get right into it.
So, let's start with the biggest question on the table. We all know things naturally want to move downhill, right? From a high concentration to a low one. But how in the world does a cell do the exact opposite? How does it actively pump things uphill against their natural tendency to create these incredibly steep concentration gradients that are absolutely essential for life? That's the puzzle we're going to solve today.
And here's our game plan. We'll kick things off by framing this incredible uphill battle that every single one of your cells is fighting. Then we'll look at the amazing machinery it uses, from the raw horsepower of ATP-driven pumps to the cleverness of coupled transport, the specialized bouncers we call ABC transporters, and finally the heavy-duty gear for bulk cargo, which is vesicular transport.
To really get why active transport is so amazing, we first have to understand the enormous challenge that cells are up against, literally every second of every day.
I mean, just look at the scale of this difference. A cell keeps almost 30 times more potassium inside than out. For sodium, it's the total opposite. And calcium, wow, the difference is just staggering. We're talking tens of thousands of times more concentrated outside the cell. This isn't just a state of being. Maintaining these gradients is like trying to hold back the water behind a massive dam. It takes constant, active work.
So, the process that makes all this possible is called active transport. The best way to think about it is like pushing a giant boulder uphill. It's not passive at all. It requires a huge investment of energy. And the cell's favorite energy currency for that job is a molecule you've definitely heard of, ATP.
Now, to get this job done, cells have evolved this incredible toolkit with 4 major strategies. We're going to dive into each one: the direct powerhouses, those ATP-driven pumps, the super efficient and clever coupled transporters, the specialized ABC transporters that kick out unwanted guests, and finally, the systems for moving the really, really big stuff. First up are the machines that burn ATP directly. You can think of these as the primary engines of the cell membrane. They create the power that in turn drives a whole bunch of other processes. Let's look at the king of all pumps, the sodium-potassium pump. Okay, watch this. It all kicks off inside the cell. 3 sodium ions snuggle into the pump's interior sites. This binding action is the trigger. The pump then grabs one molecule of ATP, cashes in its energy, and uses it to fuel this dramatic shape change, basically flipping itself open to the outside of the cell.
So now it's facing outwards, and in this new shape, the pump loses its grip on the sodium and just lets it go. But this new shape is now perfect for grabbing 2 potassium ions from the outside. And this second binding action is the key. It causes the whole pump to snap right back to its original shape, releasing the potassium inside the cell. And just like that, the cycle's ready to start all over again.
Now, this relentless cycle, which, by the way, can burn through a quarter of a cell's total energy, is absolutely fundamental to life. It creates the electrical potential that allows our nerves to fire and our muscles to contract. It builds up that sodium gradient that powers a ton of other transporters, which we're going to see in just a minute. It even keeps our cells from swelling up and bursting. And this is just one example. You've got similar ATP-driven pumps working nonstop, like the calcium pumps in our muscles or the proton pumps in our mitochondria.
Okay, so let's see how the cell gets really clever with this. If those ATP-driven pumps are building the dam and storing all that potential energy, then coupled transporters are like the water wheels that harness the energy of the water as it flows back down. It's an ingenious strategy, and it's also called secondary active transport.
You've got 2 main flavors here. In symport, a molecule that's flowing down its gradient basically pulls another molecule along with it, going in the same direction. In antiport, the downhill flow of one molecule powers the uphill movement of another one, but in the opposite direction. The key thing to remember for both is they are not using ATP directly. Nope, they're cashing in on the potential energy of an ion gradient that was already built by those primary pumps.
This is a fantastic illustration of symport in action. Okay, picture your intestinal cells right after a meal. There's a lot of glucose you need to absorb, right? The Na+ glucose symporter harnesses the powerful rush of sodium flowing into the cell, down that steep gradient we just talked about, to literally drag glucose molecules along for the ride. And this is so powerful, it can pull glucose in even when the concentration of glucose is already higher inside the cell. It's amazing.
Antiport systems are just as critical.
A perfect example is the Na-positive CCN2+ antiporter, which uses the inward flow of sodium to pump calcium out of cells. And this has huge clinical relevance, with heart medications like digoxin. By messing with the main Na-positive K+ pump, digoxin actually weakens the sodium gradient. This makes the calcium antiporter less effective, which changes the calcium levels inside heart muscle cells and alters how they contract.
Okay, our third category involves a huge and super important family of proteins, the ABC transporters. You should really think of them as the cell's specialized bouncers. Their main job is to act as efflux pumps. They grab unwanted stuff and just throw it out.
These transporters all have a similar structure. They've got two domains that bind ATP on the inner side of the membrane. When ATP binds and gets broken down, it provides the energy for this major shape change. You can picture a kind of hinge-like motion, which captures a small molecule and physically ejects it to the other side. This is a crucial defense mechanism for getting foreign and potentially toxic things out of our cells.
And this is where it gets incredibly serious in the medical world. In some cancers, the cells actually hijack this system. They dramatically ramp up their production of an ABC transporter called P-glycoprotein. This supercharged pump becomes ridiculously efficient at ejecting chemotherapy drugs just as fast as they can get in, which often makes the treatment completely useless and leads to multi-drug resistance.
So far, we've just been moving tiny ions and small molecules. But what happens when a cell needs to transport something huge, like a protein, or maybe even a whole bacterium? Well, for that, it needs a completely different strategy, the heavy machinery of vesicular transport.
This whole process works in two main directions. You've got exocytosis, which is for shipping things out. Vesicles jam-packed with cargo, like hormones or neurotransmitters, move to the cell surface and fuse with the membrane to dump their contents. And then there's endocytosis, for bringing things in. This can be pretty nonspecific, like phagocytosis or cell eating, where a macrophage might engulf a bacterium, or pinocytosis, cell drinking, to just sample the fluid outside. Or, as we're about to see, it can be a highly specific and targeted operation.
Okay, let's dive into this beautiful mechanism. Receptor-mediated endocytosis is how cells import very specific cargo with incredible efficiency. The classic example is the uptake of LDL, or what we call bad cholesterol. So LDL particles in your bloodstream bind to specific LDL receptors on the cell's surface. These receptor cargo packages then move around and cluster together in these specialized spots on the membrane called coated pits, which are lined with a protein called clathrin.
Once enough cargo has gathered in one spot, the pit starts to fold inward. We call it invagination, pulling the membrane and its receptors right along with it. With the help of some other proteins, it eventually pinches off completely, forming this self-contained little bubble called a coated vesicle inside the cell with all its precious cargo tucked safely inside. It's just a remarkably elegant and precise delivery system.
All right, we have covered a ton of ground, from single ion pumps all the way to the wholesale movement of cellular cargo. So let's bring it all together and do a quick review of the cell's amazing active transport toolkit. This table sums it all up really nicely. We've got the ATP-driven pumps, which directly burn ATP to create those powerful ion gradients. Then you have coupled transporters, which cleverly harness the energy of those gradients to move other molecules. ABC transporters also use ATP, but their main job is to export unwanted stuff. And finally, vesicular transport uses ATP to manage the heavy logistics of bulk shipping and receiving.
From the electricity firing in your brain to the absorption of your last meal, pretty much every aspect of your physiology depends on these microscopic gates and pumps working flawlessly, nonstop. It really makes you think. Given how complex and important they are, what are the consequences when even one of these critical systems starts to fail?